Hi everyone. So in this video we are going to continue with the chapter 5 of class 9th fundamental unit of life. In the previous, that is the part 1, we have studied about the cell, that is why a cell known as fundamental unit of life, its discovery. Then we have studied about the cell theory and size and shapes of the cell and other things. Okay, now we'll enter into a cell. So in this video we are talking about the journey into the cell, what will come across if we enter from outside into the cell, and what are cell wall and cell membrane, and what are their basic differences, what is diffusion, what is osmosis and all. So let us continue with this. So here we are taking a journey into a cell, that is if we enter from outside into the cell, what will come across? The first thing we'll come across is cell wall. Cell wall is found in the plant cells, in fungi and in bacteria. Remember, cell wall is not found in all the organisms, it is found only in the plants, fungi and bacteria. Then once we cross the cell wall, we'll enter or we'll come across cell membrane. Cell membrane is found in all the organisms, so whether they are plant or animal, whether they are bacteria or fungi, whether they are unicellular or multicellular, whether they live in water or on land, whether they are microscopic or large, whatever, all the cells will have cell membrane irrespective of anything. But cell wall was found only in this. So cell membrane and cell wall, two things. The plant cell, fungi and bacteria, cell wall is there as well as cell membrane. In animal cells and other cells, there is only cell membrane, cell wall is not found. Okay, once we cross the cell membrane, we'll come across a jelly-like fluid, and that jelly-like fluid is called protoplasm. And in the protoplasm, there are several things. Okay? The word proto stands for first, plasm stands for fluid. That is, it is the first fluid. Okay, it is the first fluid, and that is the most important fluid. Why we are calling it first? Because it is most important fluid. Okay, and once we enter into the cell protoplasm, we'll come across a dense spherical structure called nucleus in all the eukaryotic cell. Of course, in the prokaryotes we will not find this, and as well as whatever fluid is present outside the nucleus but inside the cell membrane, that is named as cytoplasm. So this is when we are taking a journey into the cell. So now we come across two things, cytoplasm and nucleus. Let us go into the cytoplasm. So if we take a tour down in the cytoplasm, what will come across? We will find there are a number of cell organelle present there. What are the cytoplasmic cell organelle? See, cytoplasm is between this nucleus and this cell membrane. So this is the cell membrane and this is the nucleus. Outside this entire portion, it is called cytoplasm. So in cytoplasm we will find mitochondria, on the Golgi body, endoplasmic reticulum, this is the endoplasmic reticulum, then centrioles are there, ribosomes are there, and vacuoles are there, and many other things are there. So let us see what all things we will find. We will find mitochondria in all the eukaryotic cell. We have to remember that the cell organelle, most of these are found in eukaryotic cell. Prokaryotes will have only two cell organelle, they are ribosome and centrosome. Okay, in the eukaryotes we will find mitochondria. If this is a plant cell, then we will have plastids, then we will have endoplasmic reticulum, then we will have Golgi body, we will also have vacuoles, we will have lysosomes there, microbodies are there. Of course we will not study about microbodies, and then ribosomes are there and centrosomes are there. So these are the cytoplasmic organelle which are found in a eukaryotic cell, except these two which are found even in the prokaryotes. Okay, so let me write here, they are found in the prokaryotes as well as in the eukaryotes. Okay, and remaining all are found in eukaryotes only. eukaryotes only. Now mitochondria and plastids, these are they are covered by two membranes. So they are covered by two membranes, so we will write here double membraned. These two are consisting of tube-like structures. Okay, so we'll write tubular. That's why we are grouped like this. Okay, and nothing but it becomes easy for studies, and if the question is asked, we can answer them properly. These are bag-like. They are storing something. Vacuoles and lysosomes are storing something, so they are bag-like. And microbodies, of course, it is also bag-like, so I'll continue till here. Okay, but we'll not study microbodies in class 9th. And these two are membraneless. membraneless means they do not have any sort of membrane found on them. Their body is not covered by any membrane. So this is how we are classifying them. Mitochondria and plastids are covered by two membranes. Endoplasmic reticulum and Golgi body covered, they are tubular in appearance, membranes are there. Vacuoles and lysosomes, they are having a membrane structure which separate them, and thus they look like bag-like structure, and they store something. And ribosome and centrosome do not have any membrane, and these are those organelles which are found in the prokaryote as well as in eukaryotes. Oh, sorry, this is wrong. That is, they are found even in the eukaryotes. All these structures are found in eukaryotes, and if we talk about prokaryote, only these two are found in prokaryotes. Okay, so this is clear to you, it seems. Next we'll see, this is cytoplasm. Then we have to go into nucleus. So nucleus has following parts. What are the parts which are found in the nucleus? The nucleus is covered by two membranes here, the outer membrane and the inner membrane, nuclear envelope. They will collectively form. This will have a number of pores in it. Okay, inside the nuclear membrane there is a fluid-like structure that is called nucleoplasm. This is nucleoplasm here. There are several thread-like structures. They are called chromatin network. This is chromatin network, and there is a one or more dense structure that is called nucleolus. And there are of course ribosomes also found in it. So nucleus have following part. First, nuclear envelope. Both the membrane collectively known as nuclear envelope. As I said, mitochondria and plastid also have two membranes, and here again nucleus also have two membranes. But nucleus is not cell organelle. Okay, it's a controlling unit. Inside that, whatever is found is nucleoplasm, fluid-like structure. Then there are a network of the fibers that is known as chromatin material. Then we got a dense spherical structure, one or more, that is called nucleolus. And then we got ribosomes. So these are the structures found in the nucleus. So we have studied what are found in cytoplasm and what are found in nucleus. Let's study them one by one. So very first thing is cell wall. Where is cell wall found? It is the outermost structure. This is a plant cell. In a plant cell, this is found outermost. This is the cell membrane and this is the cell wall. Light green color is cell membrane. Dark green color is cell wall. Cell wall is giving it as it's a definite shape. So you can see this is hexagonal in shape. That shape is fixed. Now you cannot change its shape because a wall is there. What is wall? This is a wall here. Okay. Now walls होती हैं, they are rigid. They are fixed. If you live in a tent, the tent will keep on moving. Anybody can press it inside or press it outside. But cell wall cannot change its shape. That is why it is called wall. It is rigid. Okay, let's talk about this cell wall. It is found in all the plants and fungi and in bacteria. The rest of the cells do not have cell wall. Then it is rigid, dead and fixed shaped. Three important things are written. Rigid means it is not elastic. It is fixed in shape. Okay, hard है and fixed shape है. And it is dead also. Dead से मतलब क्या? That is, in case of damage, it will not be able to repair itself. It will not show growth. It is not produce or develop its own. Rather, it is developed by cell membrane. So the third point, it is made up of what? In the plants, it is made up of cellulose, lignin, pectin, suberin, hemicellulose, etc. 3 things are written here, more than enough, class 9. So cellulose, hemicellulose, lignin, pectin and suberin. 5 different type of substances are there. But hemicellulose and suberin we are not written here. These are the things found in plant cells. In the fungi it is made up of chitin, and in bacteria it is made up of peptidoglycan. Okay, peptidoglycan is a substance which is consisting of proteins and carbohydrates. It is secreted by the cell membrane. Just now I said it is dead. When it is dead means it will not show its own growth and all, rather it is produced by something else, secreted by something else, taken care by something else, maintained by something else. Okay, and all that something else is cell membrane. Okay, it has several pores, and these pores are known as plasmodesmata. Okay, so these are the properties of cell wall. There are many more properties, but you all are in class 9, so that is more than enough. Functions of cell wall. What do a cell wall do? तो I said it is rigid. तो rigid है तो कुछ shape देता होगा. It provide fixed shape. ठीक है. Secondly, it provide protection. Naturally, fixed shape और wall क्यों बनाते हैं? Why we have wall around it, our house? Why? Because anybody cannot enter. ठीक है. So it is giving a protection. ठीक है. Then third, it prevent the cell from plasmolysis. Now plasmolysis is a new thing for you. If a cell is placed in a highly concentrated solution, okay, this is a ball, and we'll take water in it, and in the water we'll add small bowl, so you can add 3 to 4 spoon of salt. Three to 4 spoons. So it will become very highly concentrated. Now what you can add 3 or 4 grapes. Grapes you add, and you leave it for some time. Say for example 2 hours. So after 2 hours, what you will find, the grapes have turned. Grapes तो green होते हैं, हम green बना देते हैं. ये तो green तो दिखेगा नहीं background में. चलो, we will make it yellowish. So these are grapes. And after 2 hours or 3 hours, what you find, the grape will shrink and they will become black कर देता है. They will become like this. किशमिश बन जाएंगी. Grape will change into resins. They will lose water. A property of losing water when a cell is placed in hypertonic solution. Then what happens? The water start moving from a region of higher concentration to lower concentration. Where is more water now? Water is more in grapes, and where it is less, outside. Outside because there is more salt. So the water is now become, say for example 50%. आपने इतना पानी डाल, इतना salt डाल दिया कि now water is 50% and salt is 50%. It looks like 80. 50% इसके अंदर क्या हो गया? Salt. Where in the grapes it was 70% water. Generally living cells have 70% water. So water will start moving from a region of higher concentration, that is 70% to 50%. So it will go on shrinking. तो वो shrink होता गया, and on shrinking it will keep losing water and shrinks, and this property is called exosmosis, or rather osmosis of water. The exosmosis is called, and because of the exosmosis the cell lose water. And what happens when the cell lose water? The same plant cell which I had there, ये fixed cell wall था इसका. Cell wall remain there, and protoplasm will leave the cell membrane, cell wall, and it will shrink and become small. ये जो जगह है यहाँ ये खाली हो जाएगी. ठीक है, cell membrane will leave the wall and shrink to a smaller region. This property is called plasmolysis, but cell wall remain there only. Cell wall उसको गिरने नहीं देगा, टूटने नहीं देगा, डैमेज नहीं होने देगा. So I think plasmolysis is clear to you. Okay, it makes the skeleton of the plants. You might have seen several plants. You must have plucked a number of plants. You must have made chutney of dhania and pudina and all. Did you come across any bones in it or cartilage in it? No. Their support is actually given by cell wall to individual cell. In our body, the cells are not having individual support. Rather, the cells all are flexible and there is fixed support provided by bones. So their cell skeleton is actually made up of individual cell wall of each and every cell, which give them the entire plant a fixed shape. It provides flexibility and strength. Now I am saying it is providing fixed shape. That flexibility कहाँ से आ गया? देखिए, in certain parts you might have seen when the wind blows, the leaves they keep shaking. Okay, now why leaf shaking? If it is rigid, then it should remain like this only. It should not move. If the leaf is like this, so it will remain like this. पर वो leaf ऐसा नहीं है, वो उसकी stalk सहित हिलता रहता है. Why? Because the cells are having flexibility there. Why it is having flexibility? Because their cell wall is not continuous. Their cell walls are found more in certain corners and less at other regions. So as a result, what happens? It becomes irregular. ओके, कहीं ज़्यादा कहीं कम. तो आप एक केस लेते हैं. से फॉर एग्जांपल यू हैव गॉट वायर. आई एम नॉट सीइंग एनी वायर अराउंड मी. यू गॉट टू सिचुएशंस. यू गॉट ओके, दिस इज अ वायर फ्लेक्सिबल. अगर इसी की जगह इसी थिकनेस का मैं रॉड ले लेता हूं तो इट विल बी रिजिड. Now why this is become flexible and why rod is not flexible? क्योंकि rod का जो पूरा का पूरा metal है वो एक साथ है. In this there are several fine wires and between the 2 or 3 or 4 or 5 wires which are there inside this, maybe 8 or 16 wires, there is lot of space. So when we press it or when we turn it, there is enough place these wire get to bend. ठीक है, and since they are thin they can bend easily. So this is how the cell wall also. Cell wall is very small because the cell itself is microscopic. तो उतने छोटे छोटे हैं, but these are not regular, they are irregular. They are thick somewhere and thin somewhere. So as a result what happens, the cell wall gives them flexibility even and strength भी दे रहा है because cell wall is there तो strong तो हो गए. ठीक है, तो ये हो गए. इसके अलावा ये क्या कर रहे हैं? दे आर ऑल्सो मेकिंग द ट्रैकिड्स ऑफ द जाइलम एंड वेसल्स ऑफ द जाइलम एंड सीव ट्यूब ऑफ फ्लोएम. मान लीजिए दिस इज द सेल, दिस इज द सेल ऑफ अ ट्रैकिड. वो ट्रैकिड और वेसल की सेल जो होती है ना वो लॉन्ग हो जाती है. दे बिकम लॉन्ग एंड थिन नैरो. So now it has got its nucleus, it has got its protoplasm, etc. etc. ये सब कुछ है इसके अंदर. ठीक है, but when it get mature, तो what happens? It start losing these things. अब ये नहीं है इसके अंदर, खत्म हो गया. अभी देखिए मेरे पास इरेजर इतना पतला नहीं होता ना, इसलिए मेरे को ये करना पड़ रहा है. ठीक है, सो मैंने इसको इस तरह से मिटा दिया बैकग्राउंड के कलर के लिए. So now there is no प्रोटोप्लाज्म लेफ्ट इन इट. जब प्रोटोप्लाज्म ही नहीं है, सेल मेम्ब्रेन ही नहीं है, न्यूक्लियस ही नहीं है, साइटोप्लाज्म ही नहीं है, तो ये क्या बचा है? नाउ इट इज ओनली एन एम्प्टी बॉक्स. इट इज समथिंग लाइक दैट. यू बॉट अ टीवी. तो टीवी is come along with a well protected thermocol and then a carton. When you have removed the TV and you hung it on the wall, now you got a carton and thermocol in it. So carton and thermocol are still hard and it can still continue to provide protection. The same way, this long tube, it had number of cytoplasm and all cytoplasmic organelle, वो सब खत्म हो गए and all are dead. And on dead they become dissolved, and on dissolving क्या है वो तो खत्म हो गया. Now it become a hollow tube, and many such tube arranging on one above other makes a tubular structure, a pipeline from the root till the leaves in every plant. So that is how the water reaches to each and every cell in the plant. तो ये cell wall जो है अभी ये structure किसका बना है? This structure is made up of cell wall only. That's how it helps in this. तो cell wall के function आ गया समझ में? Provide fixed shape, provide protection from plasmolysis, chemical injuries and physical abrasions. Plasmolysis मैंने बताया, that is when a cell is kept in hypertonic solution, the water start moving from their higher region, that is in the cell, where it is 70%, to the outside media where it may be 40%, 50% or even 60%. So water will start moving from 70% to 60% and protoplasm will go on shrinking, whereas the cell wall remain there only. So that property is called plasmolysis. It also makes the skeleton of the plant to make the plant erect, and sometimes it provides flexibility at certain region. You might have seen certain branches which are very flexible. When the monkey jumps on it, the branches bends, and then when the monkey goes, the branches again come back to their original places. And the xylem, tracheids and vessels and in sieve tube, it makes the tubes. Okay, then now we'll study about cell membrane. So cell membranes are also known as plasma membrane or plasma lemma. Okay, these are the other names. Of course, these are not a property, but this is name. So sometime you may find that these are written by this structure, these terms. Okay, it is found in all the cells irrespective of their size, shape, occurrence. किस में मिल रहा है? प्लांट में, एनिमल में, बैक्टीरिया में, फंगाई में, एनिमल में, किस में मिल रहा है? कोई फर्क नहीं पड़ रहा. तो चाहे वो जमीन पे रहने वाला हो, पानी में रहने वाला हो, whatever it is, the cell membrane is found in all the cells. Third, it is living and flexible. It is not rigid, but it is living and it is flexible. And fourth thing, it is selectively permeable. That makes a very unique property. Selectively permeable. What it meant by selective permeability? Selective permeability का मतलब क्या है? जैसे आपका स्कूल है, उसमें कुछ गेट होंगे. Selective permeability is just behaving like a guarded gate where some चौकीदार हो रहा है, क्या कहते हैं, वॉचमैन is standing. So watchman or guard जो है, he will allow some people to come in and not allow certain other people. Say for example, in your school, somebody want to come in, he will ask, who are you? He says, sir, I am a parent and want to deposit fees. So he will ask which class your son or daughter is studying, and then confirm करने के बाद he may allow it. Some outsider comes, say for example, somebody want to just visit a school. Sir, हमको स्कूल देखना है, फोटोग्राफी करना है. No, you can't come unless you take permission from principal. Principal से permission कैसे लेगी? एक फ़ोन रखा है, बात कर लो. If he or she allows you, you can come in. When you want to come in, when your time, suppose your time is morning 7 o'clock, so before 6:30 to 7 o'clock you may enter. 7:30 you will come, you will be asked to stand out or you will asked to go home. Okay, so this is how. And suppose your school get over at 2 o'clock and 3 o'clock you come there, and that gatekeeper will ask, who are you, why you come? I am studying in the school, but why you come? This is not the time. So he may inquire, he may ask 10 things, he may make certain whether you should allow or not allowed, and then he'll think over it. Okay, so ये सारा काम कौन कर रहा है? ये सेल मेम्ब्रेन कर रही है. सेल मेम्ब्रेन में ask water to come in or not to come in, minerals कि आप आ सकते हैं कि नहीं आ सकते हैं. If cell already have enough of mineral, it may not allow, or if cell already have enough of water, it may not allow. तो ये सेलेक्टिबिलिटी है. कहाँ से होती है सेलेक्टिबिलिटी? ये चैनल्स हैं. ठीक है, these channels will decide कि कहाँ से इसको आना है और कहाँ से नहीं आना है. ठीक है, इट इज मेड अप ऑफ लिपिड्स एंड प्रोटीन्स. तो यहाँ पे जो येलो कलर के देख रहे हैं, वॉट एवर इज येलो कलर ओवर हियर, दीज ऑल आर लिपिड्स, एंड वॉट एवर ब्लू कलर्स आर शोन, दीज ऑल आर प्रोटीन्स. तो ये ग्लोबुलर प्रोटीन है, ये ग्लाइकोप्रोटीन है, ये यू कैन से इंटेग्रल प्रोटीन, ये पेरिफेरल प्रोटीन एंड ये चैनल प्रोटीन, थ्रू विच द थिंग्स कैन कम इन इफ दे आर थ्रू इन थ्रू, एंड दे विल अलाउ कि किसको अंदर आने देना और किसको नहीं आने देना. तो सेल मेम्ब्रेन इज अ वेरी यूनिक थिंग. Like cell wall, these are different. दो प्रॉपर्टीज पढ़ते हैं. डिफ्यूजन, यू ऑल हैव स्टडीड अबाउट डिफ्यूजन. डिफ्यूजन क्या होता है? डिफ्यूजन इज द रैंडम मूवमेंट ऑफ सर्टेन सब्सटेंसेस फ्रॉम ए रीजन ऑफ हायर कंसंट्रेशन टू रीजन ऑफ लोअर कंसंट्रेशन टिल concentration equalizes. It is a random process, no energy is required in this. रैंडम मूवमेंट ऑफ ऑल फ्लूइड पार्टिकल्स। जाएगा कौन? फ्लूइड जाएगा, ये नहीं जाएगा। ये पेन आप सोचो सर ये यहाँ पे ज़्यादा है उधर कम है तो उधर चल जाएगा, नो, solids will not go. Solid means solid can dissolve in the liquids, or solid can what you call, you have studied about sublimation. So if solids can sublimate, then they can move. But if they are strongly held together, their particles, they will not move. So solid will not move, but when they are in the liquid, when they are dissolved or when they are sublimated, they can move. Liquids can move and gases can move. These are fluid particles. So fluid particles will move from their region of higher concentration to their region of lower concentration. दिखाता हूँ कैसे होता है, एक मिनट. So if you light a sandstick अगरबत्ती, you'll see, you light a sandstick to जो भगवान जहाँ पे लगाए, मूर्ति जहाँ पे रखी है वहीं पे अपन लगा देते हैं उसको, एंड थोड़ी देर में पूरी घर के अंदर खुशबू हो जाती है. When somebody is making cooking something, वो तड़का लगाता है, पूरे घर में अड़ोस पड़ोस में सब जगह खुशबू हो जाती है. एंड when you apply deodorant, so ये deodorant है एंड यहाँ से जैसे ये निकल रहा है देखिए, एंड इमीडिएटली पूरे कमरे में खुशबू हो जाएगी. एंड डिओड्रेंट वंस यू पुट ऑन योर बॉडी एंड देन यू गो एनीवेयर, अरे सर बहुत खुशबू आ रही है, अरे यार बहुत अच्छी खुशबू आ रही है, कहाँ से आ रही है? बिकॉज दीज थिंग्स आर फ्लूइड पार्टिकल, दे आर मूविंग फ्रॉम ए रीजन ऑफ हायर कंसंट्रेशन टू लोअर कंसंट्रेशन, बट द शर्ट इज नॉट गोइंग, only the spray is going, because these are fluid, they can sublimate, they can turn into gases and they can move. So gases can move, fluid can move, solid in the fluid can move, sublimating substance can move into the gases, gases, and other things will not move. तो आप नहीं डिज़ोल्व होंगे, ऐसा नहीं कि आप यहाँ पर है थोड़ी देर में डिज़ोल्व होकर पूरे कमरे में चले गए, आप ही बचे ही नहीं. अरे ऐसा नहीं होता है यार, हम तो पता नहीं कितने सालों से जी रहे हैं, सालों से जी रहे हैं, जिए ही जा रहे हैं, बहुत साल हो गए भाई जीते जीते. ओके, सो दिस इज कॉल्ड डिफ्यूजन. तो डिफ्यूजन इज अ वेरी इंपॉर्टेंट प्रॉपर्टी फॉर ऑल द लिविंग बींग्स. There are several properties in our body which are performed by diffusion, that is the breathing. So when we breathe in, we take the oxygen, oxygen comes into the lungs, it is not oxygen but oxygen rich air comes into the lungs, and from there oxygen diffuses into the blood and blood will take it to all the body, and from there the oxygen from the blood goes into the tissues and carbon dioxide from the tissues into the blood, and वो फिर यहाँ आ जाता है और यहाँ से carbon dioxide बाहर निकल जाती है. So see, it is all diffusion. Same way the food that we eat after digestion, it is there in the small intestine, and from there it enters into the blood by diffusion from the region of higher concentration to lower concentration, that it goes to the cell. Say for example, the blood is come in this cell has got less glucose, so it will take the glucose from the blood. How? By diffusion. So diffusion is a very unique property. हम सारा जो हमारी बॉडी के अंदर काम कर रहे हैं वो सब के सब diffusion से हो रहे हैं, ठीक है. On the other hand, when we talk about osmosis, osmosis is only the movement of solvent particles from their region of higher concentration to lower concentration, and in this another important thing required is selectively permeable membrane. So osmosis takes place only in the cells. Diffusion takes place anywhere. Osmosis, only solvent particles move. Diffusion, solid, liquid, gas, any fluid particles can move. Osmosis is a slow process, whereas diffusion is a fast process. So let us summarize the differences between these two, that is diffusion and osmosis. Selective permeable membrane, diffusion में it is required, diffusion में it is not required, osmosis में it is required. Osmosis is always done through selectively permeable membrane. Diffusion can or cannot be, generally it does not require, है ना, हवा में हो रहा है, पानी में हो रहा है, when you are making tea, when you are making milk, Cadbury, chocolate, whatever you are adding into the milk, they all are diffused. That is not requiring a cell membrane, but osmosis always requires cell membrane. Material diffusion, any fluid substance, solid, liquid, gas, solid in liquid or solid in air and whatever, whereas osmosis only solvent particles move. Rate, diffusion is fast, it is slow because here an obstacle of cell membrane is there. Living or non-living, diffusion occurs in living as well as in non-living. Osmosis occurs only in living being because selectively permeable membrane is required. Now, again, Nowadays lot of RO systems, that is the water filtering system, is there, reverse osmosis. RO stand for reverse osmosis. So in this reverse osmosis we are not using selectively permeable membrane, but artificial membrane which is as thin or as capable of as a cell membrane. So here we are filling water from any source which may have certain type of impurities, and we apply a great force, high force. So when the force is applied, only and only solvent particle, that is the water, comes out of it, that is free from all salt and all other type of impurities. And that is why, so it is an artificial method. Okay, so that is why it is done, and that is not actually involving selectively permeable membrane, but a membrane somewhat similar efficiency. It is found only in living. Contact controllability, diffusion we cannot control, वो तो जाएगा ही जाएगा. But diffusion, osmosis can be controlled, because cell membrane has to choose what thing is to allowed and what thing not to be allowed. So that is how diffusion and osmosis are different on the basis of these 1 2 3 4 5 points. Okay, now let us see the differences between cell membrane and cell wall. Cell membrane found in all the organisms, whereas cell wall found only in the fungi, plants and bacteria. Composition: cell membrane is made up of lipids and proteins. Cell wall is made up of cellulose, hemicellulose, lignin, pectin, suberin in plants, and chitin in fungi, and peptidoglycan in bacteria. Flexibility: cell membrane is highly flexible, elastic, whereas it is rigid. Certain cells, they show flexibility because of the unequal depositions. And living or dead: cell membrane is always living, cell wall is always dead. It is secreted, it is maintained by the cell membrane. Pores: cell membrane does not have any pores, rather cell wall have pores, and these pores are called plasmodesmata. Location: to cell membrane, if at all cell wall is found, it is found outside, cell membrane will be inside. And in those cells where cell wall is absent, of course their location is single. Okay, permeability: so cell membrane is selectively permeable, whereas the cell wall, because of the presence of plasmodesmata, anything which are smaller than plasmodesmata can easily freely permeable, and the other things cannot. It can diffuse if it is, if it can pass through it, otherwise generally it is not. Okay, so cell wall को हम freely permeable कहते हैं, because the pores itself are so big. उससे ज़्यादा, उससे छोटी चीज़ क्या है. Okay, so cell wall and cell membrane के ये difference है. These differences you have to learn, very often asked questions. ठीक है. प्रोटोप्लाज्म की बात करते हैं. As we enter into the cell through a cell wall and then cell membrane, we come across a jelly-like fluid that is called protoplasm. It was discovered by Dujardin and named by Purkinje. Discover करा Dujardin ने, नाम दिया Purkinje. ठीक है. Everything surrounded by cell membrane is collectively known as protoplasm. So it will be having cytoplasm, and cytoplasm have all the cell organelle if it is eukaryotic cell. Prokaryote also have ribosomes and centrosome, and nucleus is also there. It is a jelly-like fluid made up of water plus organic substance plus cell organelle plus nucleus, and it is divisible into two parts, cytoplasm and nucleus. Just now we have studied, and it is living. Most importantly, it can maintain itself. Protoplasm is very unique because it can maintain its concentration. It will have 70% of water right from the time of your birth till death. It will have a certain amount of salt, sodium, potassium, calcium, other minerals are there. How much glucose is there? Everything is fixed. Neither the things are more nor the things are less. And that is why you see whenever you get your body test, this thing test done, so there is a range given. Everything is found between that range. This is because of the protoplasm, because the cell is maintaining it. Functions of protoplasm, क्या काम आ रहा है protoplasm? तो it contains nucleus as well as cytoplasm. ये इसका important function है, because everything inside the membrane तो कुछ तो होगा, जो है वो सब के सब protoplasm है. And it carries all the life functions, because the cell is carrying whatever function it is carrying out, like respiration, movement, locomotion, or what you call photosynthesis. All these are actually taking place in the protoplasm. It is living because it can maintain itself. Then it contains cytoplasmic organelle. साइटोप्लाज्म में भी पढ़ेंगे इसी चीज़ को, साइटोप्लाज्मिक ऑर्गेनेल इसी के अंदर है। चलिए साइटोप्लाज्म की बात करते हैं। एनीथिंग एंड एवरीथिंग विच इज फाउंड इनसाइड द सेल मेम्ब्रेन बट आउटसाइड द सेल वॉल इज कलेक्टिवली नोन एज साइटोप्लाज्म। मतलब प्रोटोप्लाज्म में से न्यूक्लियस को बाहर निकाल लीजिए। जहाँ हम कहते थे प्रोटोप्लाज्म क्या था, प्रोटोप्लाज्म हैड, वहाँ प्रोटोप्लाज्म was cytoplasm plus nucleus. So cytoplasm क्या हो जाएगा, cytoplasm is protoplasm minus nucleus. Now what will happen in the prokaryotic cell? There is a nucleus is not there. So वहाँ प्रोटोप्लाज्म को आप साइटोप्लाज्म बोल दो, साइटोप्लाज्म को प्रोटोप्लाज्म बोल दो। राधा साइटोप्लाज्म बोलना वहाँ सही भी नहीं रहेगा, लेकिन साइटोप्लाज्मिक ऑर्गेनेली ऐसे कह सकते हैं। तो वहाँ प्रोटोप्लाज्म इज इक्वल टू साइटोप्लाज्म। ओके, इट कंटेन्स ऑल द सेल ऑर्गेनेली इन इट, माइटोकॉन्ड्रिया, गोल्गी बॉडी, राइबोसोम, एंडोप्लाज्मिक रेटिकुलम, प्लास्टिड्स, वैक्यूल, जो भी कुछ मिल रहा है वो सब इसी के अंदर मिलेगा, नॉट फाउंड इन न्यूक्लियस। न्यूक्लियस की चीज़ें अलग है। इट इज़ लिविंग क्योंकि प्रोटोप्लाज्म वाज लिविंग बिकॉज ऑफ द साइटोप्लाज्म। ठीक है, ओके ठीक है। इट प्रोवाइड ऑल द रॉ मटेरियल फॉर द वेरियस टाइप ऑफ एक्टिविटीज़। सेल्स को जो भी काम करना है उसके लिए जो भी चीज़ें चाहिए वो कहाँ पर है, दीज ऑल थिंग्स, वट एवर थिंग्स आर रिक्वायर्ड बाय सेल टू सर्वाइव, that all are found in the cytoplasm. So it will have oxygen also, it will have glucose also, it will have minerals also, it will have other type of things also. New क्या कहते हैं, प्रोटीन्स बनाना है तो अमीनो एसिड्स विल बी देयर, राइबोसोम्स बनाना है तो जो भी चीज़ें बनानी है सब चीज़ें वहाँ पे मिलेगी। एरोबिक रेस्पिरेशन का दो, मतलब रेस्पिरेशन के दो पार्ट होते हैं, एरोबिक रेस्पिरेशन और एनारोबिक। एनारोबिक रेस्पिरेशन इन विच द ऑक्सीजन इज नॉट रिक्वायर्ड। रेस्पिरेशन विदाउट ऑक्सीजन, जो एनारोबिक रेस्पिरेशन, दैट टेक प्लेस इन साइटोप्लाज्म। ऑक्सीजन वाला हो जाता होगा दैट विल फाउंड इन माइटोकॉन्ड्रिया। ओके, so regarding osmosis, there are two type of osmosis, exosmosis and endosmosis. Endosmosis, the water move from a region of higher concentration that is outside into the cell, and in exosmosis, the water from inside the cell goes to outside. तो हम दोनों को लिखते थे यहाँ पे क्या है, that is exosmosis and endosmosis. Water from cells to outside, and यहाँ पे एंडोस्मोसिस, water from surroundings into the cell। ठीक है, तो यहाँ वाटर कैसा होगा, ये तो हो गया मूवमेंट। यहाँ वाटर कैसा होगा, यहाँ पे वाटर इज हाइपरटोनिक, है ना, एक्सोस्मोसिस, बाहर हाइपरटोनिक, एंड यहाँ वाटर कैसा होना चाहिए, हाइपोटोनिक। Here the water should be highly concentrated, है ना, पानी कम रहेगा उसके अंदर, तो पानी का क्वांटिटी कम रहेगा, वाटर इज लेस, एंड here वाटर इज मोर in the surrounding. Now what is meant by hypertonic and hypotonic? Hypertonic is that solution in which water is less, salt is more. Hypotonic is one in which water is more, salt is less. So आप ये चीज़ याद रखना, the movement of water is, ये तो आप समझ लिए, that is osmosis take place from a region of higher concentration to lower concentration, but then higher lower वहाँ बुक में एग्जाम में क्वेश्चन में आएगा hypotonic hypertonic, तो you must know how what is meant by hypertonic and what is meant by hypertonic. तो exosmosis जब हो रहा है, that is when the water is moving from the cells out to outside, तो inside the cell there is more water, outside should be less water. More water inside the cell. Cell के अंदर मान लीजिए पानी कितना है, 60% to 70% है inside the cell. Inside the cell, and surrounding में कितना होना चाहिए? It should be less than 60%. So say for example outside water is merely 40%. So water will start moving from 60% to 40%. On the other end, endosmosis में क्या हो रहा है? That resins लिए थे अपन ने. So inside water is only 30 to 40%, and the surrounding the water is 90% or above. 70%, 90%, whatever. So water will start moving from a region of higher concentration to lower concentration. So this much you must understand. Hypertonic, hypotonic की definition आपको आनी चाहिए. So that's all in this video, children. And if you really like it, so please give me a like. Okay. And if you want to share it, I would like that you share it to the other children also, other students also of your class, so that they will also come to know. देखने में बिगड़ता ही क्या है. और मैं तो कहता हूँ कि देखो भी मत. If you got time, if you don't have time, just put earphone and just keep doing your work. Definition you will learn. Definition you will learn and it will go into deep in your brain. So अगर आपके पास time नहीं है ना, subscribe the channel and please press the bell icon so that you will keep getting information regarding new videos. It takes lot of time, lot of effort, children. So please just, क्योंकि आपने पैसे नहीं दिए, आप सोचोगे अरे क्या ही बिगड़ रहा मेरा, नहीं देखूँगा तो क्या ही फर्क पड़े, ये तो आदमी स्कूल में भी पढ़ाते हैं. और don't see, education is education and it is always beneficial no matter where are you getting it from. You can get it from anywhere. The people in the past, they have even sacrificed their life in order to get their what you call education. There are a number of examples I'll tell you in the due course of videos. आने वाली वीडियोज़ में आपको बताऊंगा कि कैसे लोगों ने अपनी जान भी दी है. मैं भी आपको दो साइंटिस्ट के नाम बताता हूँ, मैरी क्यूरी और उसका हस्बैंड. व्हेन दे वर डिस्कवरिंग रेडियम, they did not know that the radiation, how hard or how the bad effect it will have on their body. They were discovering and they were finding it, अरे ये मज़ा आ गया, इससे तो मेरा हाथ भी दिख जाता है, I can through X-ray and this, and it emit light in the night and it is good thing. They used to keep some of these in their rooms and it used to glow in the night and they used to enjoy. But they did not know that the radiation which is coming out of it, that caused cancer in them, and both of them died. But in course of studies, so that today we come to know what is this. ठीक है, तो एजुकेशन तो कहीं से भी हमको मिल रहा है, एंड प्रैक्टिकली एवरीथिंग देयर इज सम साइंस. हर चीज़ में साइंस है, तो आप कोशिश करिए. और जब आप ये करने लग जाएंगे ना, जब यू विल स्टार्ट एक्सप्लोरिंग कि हाउ माई साइकिल इज़ मूविंग, what all processes are involved in it, or how a fan is working, or what all processes are involved in it. जब आप ये करने लग जाएंगे ना, तो आप you will do number of new things. That's all in this video. I don't want to just make the video too long, and see you in the next video where we'll discuss about the cytoplasmic cell organelle, mitochondria and plastids, and endoplasmic reticulum. 4 we'll do in the first video and the remaining 4 we'll do in the next video, and then differences between the plants, animal, prokaryote, eukaryotic and cytoplasm. Altogether 5 or 6 videos will be there, so the video will be